All right. I'm coming. All right. Hi, everyone. So I'm Abisola from Case Western Reserve University. And this is an experiment I carried out last summer, trying to look and test whether Trichoderma species can actually enhance rhododendron performance under pathogen stress. So this experiment is based on prior evidence of soil biotic community enhancing rhododendron survivor under pathogen stress. So one thing I did here was to manipulate Trichoderma, and I chose Trichoderma as well because from the high throughput sequencing that was done on prior experiments, we found Trichoderma, which is a fungal genus, to be abundant in those soil fungal community. And based on the fact that Trichoderma biocontrol agents, and they also have like plant growth promoting effect. So we decided to test whether they also have this same effect on rhododendron species. So for this experiment, we chose like 3 different rhododendron species based on their susceptibility to this root rot pathogen Phytophthora. So we have the maximum mole and Schlieppenbachii, and from the results, so we found out that it's a greenhouse experiment, and we found like a reduced disease symptoms for those that are treated with this trichoderma, actually for 2 out of 3 species we use, which is maximum and mole. So we can see like there's actually like species-specific responses, but we don't know the mechanisms underlying that yet, but we can see like those trichoderma treated ones, they actually display a much lesser disease symptoms compared to the controls. And we also look at the leaf damage as well, and this damage was significantly reduced by trichoderma species compared to control as well. And we also saw like an increase in the shoot biomass. So, however, all these things, the mechanism by which trichoderma carried this out is unclear yet, because they can actually induce systemic resistance in this plant. They can also produce like inhibitory compounds like peptide bonds that help to inhibit phytopathogens as well. So this is what we did, and currently we are trying to look if this trichoderma can actually shift the native soil fungal community in rhododendron species. So we'll be looking at its effects, the trichoderma effects on the native soil biota as well, and trying to see this interactions between the general soil fungal community with trichoderma as well as the rhododendron species themselves. And also we are looking into test like this trichoderma effects on like a wide range of rhododendron species and try to quantify their like species specific responses. Yeah, so I would like to take questions now. Thank you.
While we're taking questions, I'm going to put the link to the posters in the chat, so you can head on over to the posters. If you don't have a question immediately, you can look at the posters and ask a question on the poster site, or we can take a question now through this same format that we've been using. I have a question that's a methods question. How do you get trichoderma?
Okay, and this trichoderma were actually bought from American Type Culture Collection. So we want to make sure like we get the specific trichoderma species, which is harzianum and asperellum, because these species were found to be part of the trichoderma species found in the general fungal community from the IEEE sequencing that was carried out. Yeah, so we bought that.
Great.
Yeah.
We have a question from Dylan Nakaji Conley. Dylan?
Hi. So were there any, I guess, morphological or physiological traits about these different species that you know of that you think might have to do with these differences in response to the benefit of trichoderma?
Um, yeah, I believe there will be morphological differences which we didn't really look into. But one thing I know about the rhododendron schlippenbachii is if it's like under stress, it tends to like shed its leaves. And when the condition is like favorable again, they tend to like pick up and grow more, right? And based on these different species as well, the maximum is evergreen and mole and spermaca, they're like deciduous. So I believe their morphological differences could also contribute to their responses to this trichoderma, as well as their root exudates as well. Yeah. Question.
Wonderful. Thank you. I think we need to move on to our next talk. As I mentioned, don't forget to visit the poster session over there and you can ask more questions.